So today I'm going to be talking about hormonal control of implantation and early embryonic development. So basically we're at the part where an egg is fertilized, it becomes a zygote, it begins mitosis as it makes its way along the fallopian tube to the uterus, and that's where it's going to settle for the remainder of the gestational period. So the dividing embryo takes 4 or 5 days to move through the fallopian tube into the uterine cavity. And under the influence of progesterone, smooth muscle of the tube relaxes and transport proceeds slowly. By the time the developing embryo reaches the uterus, it consists of a hollow ball of about 100 cells called a blastocyst. So some of the outer layer of blastocyst cells will become the chorion, which is an extra embryonic membrane. It will enclose the embryo and that will form the placenta. So the inner cell mass of the blastocyst will develop into the embryo and into 3 other extra embryonic membranes. So we have the amnion, which secretes amniotic fluid, and that's where the embryo floats. Then the allantois, I'm not 100% sure how to pronounce that, which becomes part of the umbilical cord that links the embryo to the mother. And then there's the yolk sac, which degenerates early in human development. So implantation of the blastocyst into the uterine wall normally takes place about 7 days after fertilization, and the blastocyst secretes enzymes that allow it to invade the endometrium, like sort of like a parasite, like burrowing into its host. As it does that, endometrial cells grow out and around the blastocyst until it is completely engulfed. So as the blastocyst continues dividing and becomes an embryo, cells that will become the placenta form these villi called chorionic villi. They penetrate into the vascularized endometrium, and enzymes from the villi break down the walls of maternal blood vessels until the villi are surrounded by pools of maternal blood. So the blood of the embryo and of the mother, they don't mix, but nutrients, gases, and waste exchange across the membranes of the villi. Many of the substances move by diffusion, but some simple diffusion, but some, such as maternal antibodies, have to be transported across the membrane. So the placenta continues to grow during pregnancy until by delivery, it's about 20 centimeters in diameter. And the placenta receives as much as 10% of the total maternal cardiac output. So that huge blood flow to the placenta is one reason for the sudden abnormal separation of the placenta from the uterine wall, and why that's like a medical emergency. So moving on to the hormonal control, as the blastocyst implants into the uterine wall and the placenta begins to form, the corpus luteum nears the end of its 12-day lifespan. Unless the developing embryo sends a hormonal signal, the corpus luteum disintegrates, and progesterone and estrogen drop, and the embryo is flushed from the body, along with surface levels of endometrium during menstruation. So the placenta secretes several hormones that prevent menstruation during pregnancy. So the first one that I'm going to talk about is human chorionic gonadotropin, which we're going to call HCG. So the corpus luteum remains active during early pregnancy because of HCG, which is a peptide hormone. It's secreted by the chorionic villi and developing placenta. HCG is structurally related to LH, and it actually binds to LH receptors. Under the influence of HCG, the corpus luteum keeps producing progesterone to keep the endometrium intact. By the 7th week of development, the placenta, however, has taken over progesterone production and the corpus luteum is no longer needed. So at that point, it finally degenerates. HCG production by the placenta peaks at 3 months of development, and then it diminishes. So there's also a second function of HCG, and that's to stimulate testosterone production by the developing testes in male fetuses. Fetal testosterone and its metabolite DHT are essential for the expression of male characteristics and for descent of the testes into the scrotum before birth. HCG is the chemical detected also by pregnancy tests. So it can actually induce ovulation in rabbits. So years ago, rabbits were used for pregnancy testing. So if a woman thought she was pregnant, they would actually inject her urine into a rabbit. Obviously, this is not something we do anymore. Now, with modern biochemical techniques, women can perform their own pregnancy tests in a few minutes, but with detecting this hormone. All right, the next hormone we're going to talk about is human chorionic somatomamotropin, which we're going to call HCS. This is another peptide hormone. It is produced by the placenta, and it also has another name, which is human placental lactogen. So this hormone is structurally related to growth hormone and prolactin. Initially, it was believed to be necessary for breast development during pregnancy and for milk production, which it probably does contribute to lactation, but women who don't make it during pregnancy still have adequate breast development and milk production. So a second role is the alteration of the mother's glucose and fatty acid metabolism to support fetal growth. So maternal glucose moves across the membranes of the placenta by facilitated diffusion, and it enters fetal circulation. So during pregnancy, about 4% of women actually develop gestational diabetes mellitus, and that is elevated blood glucose levels caused by insulin resistance, which is similar to type 2 diabetes. The cause is kind of unclear, but basically after delivery, glucose metabolism in most women returns to normal, but these mothers and their babies are at higher risk of developing type 2 diabetes later in life. All right, now getting to estrogen and progesterone. Estrogen and progesterone are produced continuously during pregnancy, first by the corpus luteum under the influence of HCG, and then by the placenta. With high circulating levels of these steroid hormones, feedback suppression of the pituitary continues throughout pregnancy, which prevents another set of follicles from beginning development. So during pregnancy, estrogen contributes to the development of the milk secreting ducts of the breasts, and progesterone is essential for maintaining the endometrium and also helps suppress uterine contractions. The placenta makes a variety of other hormones, including inhibin and prorenin, but the function of most of these remains unclear. So these were the main hormones that I wanted to speak about in my presentation today, and I hope you learned something about hormonal control of implantation and early embryonic development. Thank you.